Hey, good morning, Mike.
Morning.
How are you doing?
Good, good, good. I was out sun tanning yesterday and the back was too above, but the sun was really warm. So I got my shirt off and got, got some vitamin D built up.
Perfect. Got a piece of tinfoil out and got the reflection going.
Uh,
We
well,
can't wait for spring. That's the way we are, right? As
well, it won't
soon
take
as we
long.
get
It's
in.
only eight above this week, so there's not much snow left.
Oh, well. Yeah, we do have some on the ground here in Manitoba, but yeah, it's supposed to warm up as well. So, yeah, another spring, or second or third spring this winter. Yeah. Well, we're going to start, Mike, here in a minute or so as people are coming on. Just to let everybody know, we will be recording this uh, conversation, so it'll be available to the attendees after. And we'll send them a direct link, and then we'll also probably push it out through our social channels as well. Our goal is to try to keep it within an hour if we can. If we go a bit longer, bear with us, just because we're probably in deep conversation over some topic. But yeah, we don't want to keep you too long here this morning. So yeah, we will get started here. Good morning, everybody. Craig Davidson, Taurus Ag Marketing, president and founding partner out of Manitoba. We also have Mr. Mike Dolinkski with us, our director of science and innovation. You want to say hi, Mike?
Hi, guys. Nice having you on this morning.
Yeah.
And
Little
girls,
housekeeping.
and girls.
Yes, girls and guys, you betcha. Little housekeeping. We do have CCA credits available for this conversation today. There is a QR code you can see on your screen, but that you cannot take a picture of that. You have to go on to the CCA app and take a picture of that to download to get your credits. So you'd have to download the CCA app first, and then find the trace genomics presentation from Taurus and go about it that way. So yeah, well, we'll get rolling in the essence of time. We're going to start today with a little background on Taurus. Obviously, I mean, we've been around for a long time, 23 years in business. Our focus is agronomy and fertility, and here we are today back into what we would call agronomy through genomics and leveraging, our previous experience in analytics. But for the most part, I mean, we're focused on agronomy when we look at fertility, biological inoculants, primers and foliars, and even nitrogen management. And our pillars today are full, and we continue to look for new innovations that help growers be better at what they do. Obviously, the word efficiency hasn't gone out of style. The word sustainable is a bit of a buzzword today. We feel everything we do fits both of those in spades. And our conversation today on trace really only takes that further down the path when we look at more information, more data to make more informed decisions. That's really what we have built our business on, and that's our bread and butter, to allow growers to be better at what they do. But for the most part, I would say over the years in our business, and obviously, having Mike in the fold for the last number of years, I think maybe one of our greatest assets is being inquisitive and asking the hard questions and trying to figure out the whys behind the hows. And so when we look at something new, we tend to have a lot of questions. People probably get sick of us with the amount of questions we ask, but all we're trying to do is figure out, what is the science, where is the fit, where is the value, and how can it benefit growers to be more successful in entering 2024, what could be a very tight economic state for the production of agriculture. And when we look at something new in science, we're going to ask questions. But in the context of trace and trace genomics, if you ask this question to yourself and you have no idea what we're going to talk about if you haven't heard of trace. But these are some of the questions we asked in the early days, and this is some of the things that they brought to us. What if, as a grower, as a consultant, as a retailer, you could identify core pathogens in your soil that could affect, say, something like wheat before you planted or before you treated the seed with a fungicidal seed treatment, and you had a good indication of what your core pathogens were that could potentially cause harm to that seed. Would you want to know that? Would you actually want to know that if if that information was available? That's a question we had. What if before you grew your wheat, you knew the pathogens in the soil, and you knew what fields potentially had the highest concentration of Fusarium head blight as a pathogen in the soil? Would you then be more cautious or be more apt to be proactive in the way you manage those fields heading into the summer months or in the heading of that plant or that crop? What if you were sitting down right now in the middle of winter and you're building out your rotation for the coming season, it's 2024, and you had a better handle on pathogens in your soil that you really had no way to manage other than rotation? Would you actually alter your rotation if in fact you knew, and say in this case, you knew the fields that had a background level or the fields that had the highest concentration of a disease like Phanomyces, and you were trying to figure out where should I be putting my peas in 2024 or 2025? Would you look at that decision-making process differently? What if we are being governed to manage something like nitrous oxide, but I took all of my fields on my operation or across my geography, and I had a better indication of the fields that were more prone to nitrification, that in turn could be more susceptible to nitrous oxide emissions? What if you had this information? Could you potentially be smarter than a government program and then leverage agronomy to best manage your inputs, like something like nitrogen, to protect it from potential loss? If you had this information, would it change the way you think? What if there's a new disease that came along that had everybody a little bit scared, but nobody really understood much about it, and because it's new, there's really no control mechanism other than extending a rotation? What if you had this information before you sat down and planned out a rotation? Would you potentially change the way you build out your recommendation for rotations on your farm, or managed a crop like canola a little bit differently? If all of a sudden you know that this disease is here and I don't want to propagate it or build it any more than I currently have it in my soil, would you go about it differently? Well, today, when we look at those what if questions, and we look at something like DNA that has been around since, it was discovered in 1953. Mike, who were the guys that actually brought this forth?
No, it was discovered long before that, but nobody really knew what they discovered. But in 53, Watson and Crick actually published on sort of what the function of DNA was.
Okay. So we're talking 70 years ago. 70 years ago, and it took them close to 20 years to actually start to sequence that DNA. And 20 years later, in 1990, they started to look at the human genome with the idea that they could actually dissect the living aspect of the human body. Well, that actually took 13 years. So you look at this roadmap, and here we are, 50 years of work to get the Human Genome Project completed. And you said, that's a long time. I mean, that's a half a lifetime or longer for most people. And then now, from this point forward, you say, well, this is where technology kicks in, and you start to see the speed and scale of advancement in something like DNA and DNA sequencing, and the user friendliness that has come along in the last, call it 20 years, that's allowed this to scale quickly. And for the most part, a lot of this work, and you could correct me if I'm wrong here, Mike, but a lot of this work in the last 20 years has really been focused on the human genome. Is that correct?
That's about right. That had to be the priority, and there was billions of dollars spent on doing that. And in the beginning, they're sort of very, very slow. Today, we can do a human in a day. That's what's been amazing. The speed at which we can do is what's called the new generation sequencing machinery, can run really fast and do massive numbers simultaneously.
Yeah. So in our world, we are fortunate to be able to work with Trace Genomics and Dr. Purnima Peresmas-Warren, who's the president and co-founder of Trace. And she actually looked at what was going on in human science and said, what else could we apply this innovative technology to? And obviously with her background, with some connection to agriculture production, she said, what about agriculture? Is there a fit leveraging some of this technology in agriculture? And so that was the beginning of Trace Genomics. I have this picture from a couple weeks ago in Regina. She came up and presented at the Convergence conference, and they did an amazing job. But what I really like about the picture is we got her culturized in Saskatchewan pretty quick. You can see if you look close, she's drinking a Pilsner. And she's a solidified Saskatchewanite. So yeah, she's not, every time she comes up, she knows what the locals will do. So maybe talk a bit about what Trace is working on and how we actually look at this technology when we look at the human genome. Mike, what is metagenomics?
Metagenomics is sort of a term used that beyond genomics. And what we can do with metagenomics is we can take a soil sample, extract all the DNA, and then running through the new technology with Illumina machinery, we can identify all the sequences in that soil sample. Now, all the data that we present going forward is based on the DNA from the microbes, from everything in the soil. Just to give you, we talk about billions of organisms. Just to give you some idea on the quantitative sort of values, in every soil sample, we get an average of about 11,000 gene sequences that could be representative of the species in that soil sample. So up until this point in time, we really had no idea here in Western Canada what we had in our soil until basically the 1st of January almost, when we were having samples processed by Trace and development of benchmarks. And I'll get into that in a minute. Benchmarks for what do these values mean? Because the values presented are in parts per billion. And the sequences that are there represent diseases. But if we got 11,000 species or sequences, we only know a few of them. You can't identify that organism unless you have sequenced it. So we have 250 to 270 diseases of crops that we have sequenced. So we know when we search through the DNA database, which is done sort of by artificial intelligence, we can pull out those 270 diseases, but they're all based on DNA. So if you take a sample from a field, and we'll show you this in a while, and then you can just switch crops, all the diseases of wheat, canola, barley, and I think, Craig, it's 70 or 100 crops that we now have that we can use this information for. So that's where we're going. We can take that now. One last point is, because we don't know the majority of what the sequences identify, we can look at the functions. For example, if we know that there's a gene, and I always use nitrogen fixation, for example, a NIF gene, we can search that database and look for the NIF gene because it's got a specific sequence. If that organism, which we really don't know what it is, has a NIF gene in it, we can speculate that that is an organism that can actually fix nitrogen. So that's what metagenomics is all about, taking all the DNA, that includes all the plant DNA, and we got to get rid of that scrap material to then identify the organisms that are there, and we identify them all, not just one or two or a couple, all of them. And that data is there forever. It's stored at minus 70 degrees, an actual sample, and the data is stored. So we can go back in time. For example, if we start now, in 10 years, we can go back in time. That data will be there, and we'll be able to identify the changes that have occurred in that soil. That's sort of, in a nutshell, what we're talking about.
Yeah, I would explain it as you send in a sample of soil, and they can test every single strand of DNA that's in that bag. Now, they may not know what every strand is. If they have the genetic markers, they can identify it, but the data is never lost, is what you're saying.
Yeah, never lost.
Yeah. So how would you relate that to the advancement of the exact same technology, like we said, that was basically evolving through human science? Where do we see that today? Because I know you, Mike, are a very passionate individual connecting humans to plants. And now we're going to connect humans to soil and the biology of the soil and the biology in a human. How do you see that? How do you see this technology benefiting society as a whole?
Well, it's going to benefit society as a whole because the soil is one of the biggest reserves of all the microbes on Earth, you know, between the oceans and the seas and the soil. And the soil maintains the largest number of them. So a lot of the stuff that we're getting has originated in our gut microbiome through what we've eaten. And really, for me, the big advancement in human health has been recently in response to the Human Genome Project, the gut microbiome analysis, and that's been done by metagenomics. Because we now know that in the gut, we have more DNA than our entire body, probably by a factor of 10 to 100 times more DNA in our gut. And it's those microbes in there we now know are really responsible for the cycling of our nutrients, just like the microbes in the soil are somewhat responsible for the cycling of nutrients in the soil and the organic matter and releasing those nutrients to plants and that relationship between the plants and the soil. So there's a concept developing now, it's called the One Health concept, where we know that the animals and the plants, which humans basically eat, are involved in human health. And soil is the basis of all of that, because without plants, we don't have the carbon that is essential to feed to those microbes down in the soil, rhizospheres and so on, that are really the source of energy for them and make those relationships with the plants. And then we get the benefits of those relationships from the soil in humans, as well as all those microbes that we intake. So that's kind of the way it is. And we're now starting to get to a point where we're understanding the human microbiome and the relationship to the brain, but we're also understanding it in terms of all kinds of other functions in our human body. We're just entering that same phase with soil right now in Canada, for sure. Maybe they're further advanced in the US because they've been at it a little longer, but that's where we're going, where the Human Genome Project has taken us in human health. We'll be able to go there and look at how these organisms are involved in both plant and animal health as a whole.
Yeah, so pretty cool in a sense that it's not a new technology for agriculture. It's a technology that transcends, I guess, the living aspect of Earth, whether it's humans or plants or soil, and we're leveraging that all at the same time.
All at the same time.
For that.
There's a general trend to try. There's actually a concept to try and identify all the DNA on Earth. So we have it recorded. Okay. Yeah, we have it recorded, but that's another matter.
Okay. So if we look at trace, specifically trace genomics, and what Dr. Praneem has been a part of since 2016, really it was adopting that innovative technology that was being leveraged in human science and started to bring that to the soil. So we're 8 years into this, I guess, journey that Trace has been on. And, in the early days, obviously, the microbiome diagnostic capability was limited. Obviously, 5 crops and a handful of diseases. As time progressed, I think she would say in the early days, to fully sequence a sample, it took weeks for the machine learning, the machine technology to sequence a sample and identify the DNA within that sample. Whereas as we sit here today in 2024, that same sequencing event is a matter of days, 5 days, 6 days to fully sequence a soil, which is amazing how it's come from weeks and weeks to just a handful of days, which is almost as quick as today what it would take to do a chemistry analysis on a soil that you send in. And it just so happens in our case, as we stand today, Trace is doing both chemistry and biology and carbon because we want to leverage the interconductivity between those analysis to better understand the whole profile, the whole soil, and how that can work or not work for a plant that we're trying to grow. So as we sit today, it's quite robust. It's a commercially viable product that gives us an insight into the biology of the soil. It gives us insight into fertility and nutrient cycling. We'll talk about that. We look at things like seed selection, seed treatment options, in-season disease management. So the idea of chemistry, and that's where a lot of us are today. I mean, and it's an extremely valuable tool in making good decisions from a fertility perspective. Today, when you look at a Trace Complete analysis, I mean, it is multifaceted and multi-dynamic in its ability to better understand the soil. And up until now, without DNA sequencing, how do you actually test or measure the living soil from pathogens to beneficials to organisms that form relationships with plants? And yet today this changes. You said pathogens, Mike, they're doing up to 225 plus pathogens today. We're covering over 70 different crops. And so for us here in our market in Canada, I mean, the dynamics of that give us an insight to the future of agriculture. And I always say disruption takes years, not days or weeks. This is probably, we're at the forefront of changing the way agriculture operates and how growers make decisions, how agronomists make decisions. And in the early days, it's scary. It's kind of scary to say, holy cow, this is overwhelming, to go from just chemistry to now you have all this other detail from our soil. How do we actually do it? Like, how do we actually figure out how to make use of this? And that's probably the common question we've had with people that we've worked with in the first few months to benchmark out the Canadian market is how do we make use of it? Where is the actual value? And so that's what we'll get into a bit here, is to kind of say, well, this is, what can you do? What can you do once you have this information? Part of it for us is really not just the technology in DNA sequencing, it's actually the investment in the technology of the interface that Trace has put forward in TraceView that allows us to visualize and leverage this data in a user-friendly format. And that's probably, I mean, I'm not going to be as in-depth as Mike when it comes to understanding the living soil and the impact that these organisms have on a plant and the interrelationships that are formed. But if I can actually make use of it in a simplified view, to me, that's just as valuable. So we do give Trace Genomics credit for actually putting together and investing in the AI aspect of leveraging all the data that comes. And I believe, correct me if I'm wrong, Mike, but for every sample, there's roughly 5 gigabytes of data.
That's right.
Yeah.
You can imagine if you've got 11,000 strings of DNA or species potentially. That's a massive amount of data that is accumulated, and the cloud space is huge. Just to maybe broaden out the capacity, the company has two sequencers that run 384 samples in a batch that take about 3 days to run them through. So they'll actually sequence those 384 individual soil samples and identify all the DNA sequences in all of them over that 3-day period on the run. And before that, there's what's called the pipeline. There's the preparation and so on. So it takes a while, and the energy and storage capacity is just beyond me. Plus, their cold storage at -70 keeps an actual real sample of that DNA forever if they have to go back to it. Give you some sort of idea on the capacity.
Yeah, and when we look at it, we do think that there's value in the full chain on the agronomic side, whether you're an agronomist or consultant, a retailer or a grower. I mean, ultimately, at the end of the day, all that matters is that there's value for the grower when it comes to figuring out ways to find more production on that field or on that acre. How do we reduce the potential risks that could be coming our way, or potentially affecting the crop that we're growing? When we look at the sustainability side of it, there may not be, we are fortunate in Taurus that we have some things that are directly tying humans to production of food. But I think this fits that category as well, where, all of a sudden we could say, yes, we're using tools in our toolbox currently, but we don't know for sure if we're got the right tool for the crop we're growing, or say there's a set of pathogens in that soil that we didn't know for sure what they were. Now we do. So we select the right tool in the toolbox. We pick the right seed treatment to protect from disease. Well, to me, that is absolutely leveraging sustainability, because we're not building resistance. We're only protecting against something that's currently there or at high concentration. And, the ideas of, say, something like we're going to be forced to be better at nutrient utilization. Well, what if, in fact, now we can best manage nutrient loss where it actually is potentially at risk because of organisms in the soil that convert nutrients from an unavailable form to an available form or back to a gaseous state. So your ability to be more defined in your decision-making process totally changes. And to me, that tells me you're more efficient, you're using products more effectively, and you're not wasting products that could actually build resistance in the long run. That's what we need in agriculture. We just haven't had those tools to make those decisions till now.
That's right. And, we on the soil side of it, basically the history of our soil science has been soil chemistry. The role of microbes is only recent, where we knew they were involved, but we didn't know, the implications, and some of the testing has given us a little bit of vision into how these microorganisms expand and grow and how we can maybe measure them. But for the first time, we can actually specifically identify those organisms and track them through time. We're in the early stages, and talking to people in recent times, everybody wants to know how we're going to use this and what is going to be the long-term ramification. And I don't know, I wish I was 40 years younger to see that, because I think it's only through the next few years where we develop some databases for Western Canada and Ontario and the entire country, for that matter, and then be able to look back and utilize that data to make decisions. But if you get a sample that has a disease, you at least know where to look, which fields to look, and from a basic agronomy point of view, make decisions to deal with those. Because if we don't continue to make reasonable decisions about the application of our technology in agriculture, it would appear that governments will clamp down on us and restrict them. And we have to be in a position to say we are doing a really good job on this. So that's where I see the long-term benefits of this. And it doesn't matter when you start. If you start today, if that data will be there forever. And every time it is you turn on your portal and you look at your data, it's updated automatically. So it goes back and it builds on the benchmarks, which I'll get to in a minute. But you want me to take over here and take a look at this, Craig?
Yeah, I was just going to make a comment. Yes,
Okay.
obviously, you posed the question, start whenever. I'd say that's fair. We're coming in at a time where Trace has been at it for 8 years. The fact that they have proven out their technology in the U.S. and it's commercially being used on a tremendous amount of acres with progressive thinking growers and agronomists. For us, we're kind of saying, well, we've spent the last few months benchmarking, trying to get our baselines established of where our organism content is, call it on a national level. We're trying to bring that down to a regional level to be more specific to your growing conditions. But you'd say when you really start to see the value of knowing more information, you'd say, why wait? If you wait, what's the value of waiting? What's the value of waiting? Well, at this point, we're kind of saying, I'm not sure. Why would you wait when when we can figure out where you're at now, and the fact that you can backtrack to that sample in the future and say, well, you know what? We didn't even know verticillium stripe existed in 2024. They found it in 2027. I can go back to my 2024 sample and say, yeah, well, you had it in your field, though we didn't know what it was at the time. And this is what's happened over 3 years or 4 years. You've built that pathogen to a higher concentration, causing more concern. So it's my question is, why wait? Yes, it's new. It's a lot of information. You got to gain comfort. But when it comes down to finding something that allows you to make a positive decision, we'll go through it here in the next 20 minutes. But there's lots of things you can check off that are probably relevant to what you're trying to decide on today.
Yeah, I guess I should qualify when I said why wait. I mean, I meant start now.
Yeah. Okay.
It doesn't matter, if you get the data, you got it now. It'll be there forever. It'll be reviewed on an ongoing basis. The benchmarks will be changed as we get more data for Western Canada. Yeah, I meant don't wait. You don't want to wait, because it doesn't matter when you start. If you start it now, we will be learning this over the next umpteen years, probably for generations. This data will be there. If your family is going to continue farming that land, that's going to be value. Even just think about this, folks. If you have this data on your farm, on your quarter sections or sections, that brings value to the sale if you're going to sell your land. Think about that. What's that going to mean to buyers, to your next generation, in terms of understanding this soil? Just think of what's happened in the last 50 years. We're starting basically now. What's going to be the situation in 50 years from now when your son or your daughter or whoever is going to be farming that land? So yeah, start now. You'll find out where to look. You'll find out what diseases you have. You'll make better decisions about fungicides if you need to use them. All kinds of decisions. Anyhow, I wanted to qualify that. But we're now moving into a stage here with we're recognizing that plants and microbes in the soil have a direct relationship through the exudation that the plants provide for the microbes, which are the carbon source for many of those microbes. And that relationship between the microbes and the soil and the plants is now becoming, I guess, commonly spoken of, and we're getting a handle on it and how that's all cycling in that system. Give me the next one, Craig. I just throw this in because part of the component of the soil sample, which is a 0 to 6 inch sample, we find that size of sample gives you a good indication, and you're probably looking at 2 or 3 samples per field, and you can base those samples on some sort of a criteria, whether it be a high, middle, and low slope, yield data, whatever satellite data. You can get some values, and it's probably best take about 3 samples in a field, simply because if you just have one, you don't know how that's relating to anything else in your field, whether that be low spots, high spots, saline positions or whatever. So this comes automatically with it, and it's somewhat necessary, or I should say really necessary, because if you're going to look at how microbes are influencing the cycling of phosphorus, how microbes are influencing the cycling of the nitrogen in your soils, you're going to need basic chemistry on those samples.
Yeah, so if we look at the test, when we talk about DNA sequencing, essentially in our world, the commercial test today will be called Trace Complete. Trace Complete also includes the chemistry aspect of the soil, because you are actually, in some of the nutrient cycling, Mike, you're actually leveraging chemistry with the biology. Is that correct?
You bet, you bet. And I was down at the facilities in Ames, Iowa. This is where the lab is. And they're set up really well. This is all done. It's all done by computer in terms of data recording. So it's very, very accurate.
Yes. So this is what the market expects. Obviously, this is where, meet the market where they're at currently. The market is doing good chemistry work through various labs that are across North America. And essentially we're saying, yeah, we're going to continue that on, or you can use your own lab for chemistry if you want. But in all reality, the true value of Trace Complete when we talk nutrient cycling is combining chemistry with biology. And we'll show some of those examples.
And from my perspective, because I look at this long term, is if we have, because all these sites are GPS marked, we can go back to them in time and resample them. So that's really important, because I think in the long run, we may be able to draw some conclusions about the levels of nutrients in the soil and the way that disease organisms and beneficial organisms behave over time. That's why having, for the first time, both the nutrient levels, the organic matter levels, and the disease and beneficials in the same sample gives us probably the best database we could ever have for that field. Okay.
Yeah. Talk a little bit about the idea of benchmark, and this is what we're currently doing, is basically establishing a baseline for our market. Call it, right now they have a national benchmark because of the work they've done in the US. We're trying to establish, call it regional benchmarks, either Western Canada or Eastern Canada or provincial or even tighter than that. What do the numbers actually mean, Mike, or how would you define, the percentile ranges that they create for anything from seed selection to in-season disease management and things like that?
Almost everything we do is on a bell curve. And you can see here at a low level is 25 percentile, the high level is 90% percentile. And that just gives you some confidence in what those numbers mean. So when we go forward, you'll see that the graphs are going to be shown in colors. Green means that it's a low level of inoculum, because we're looking at, say, the DNA. It's a low level of DNA, which implies that it's a low level of inoculation. What's the word? Inoculant. There you go, the inoculant for that organism. So that implies that, well, that's a really low risk. If we take a whole bunch of samples across Western Canada, which is part of the benchmark, we've collected somewhere between 400 and 500 samples from Western Canada. So we get some idea on what is high for a disease and what is low. Because if we don't give agronomists and farmers some idea on what it means, it's just a bunch of data. So the simple interpretation is, if it's below 25%, that's a low level and may not be of much concern. If it's in this medium zone between 25 and 90, well, we don't know what the environmental conditions are going to be in any of these spots. And that's why the agronomist and the farmer, with their local knowledge, is really key to the understanding and making the decisions. And we'll get into it a little bit more when we look at the disease cycle. Now, once you get to 90 percentile, which is sort of the benchmark that you'll see running across the graphs, then we know we're in a high level of risk. It's somewhat of a risk analysis, in my point of view, of saying, I better really watch that field, or mine better take some action. Or if I'm looking at a summer program, I know my fusarium is really, really high in the soil, so I'm going to really watch that field over another field. It'll vary because the infection is related to environmental conditions as well as the inoculant. But those are what those numbers are going to mean. And here's where we go. So in this case, we now have this part. We've got the pathogens. We know the pathogens that are going to be there. You on the farm or the agronomist know the host, you know the resistance and tolerance to those, and you know what environmental conditions are required for that pathogen in that host combination to in fact lead to disease. So the assessment is still going to be really important at the local level. And I think, Craig, you've got a lot of experience, and we know that, for example, sclerotinia and canola, I mean, I always talk about canola in a large sense because it's such a big crop in Western Canada. We can have a lot of inoculant if we don't have the right humidity conditions and we don't get infection, or if it comes at the wrong time. All of these diseases have certain criteria in which they get infection. Now, the other beautiful thing that has happened with technology besides this is that we now have mechanisms to monitor these diseases if they're in the air as well, because that's the other part of the spectrum. So if you tie in the environmental conditions for something like soil moisture monitoring, leaf moisture monitoring, those kind of things that we're now developing, along with mechanisms to monitor spore movement, as we do with fusarium and sclerotinia, and we know what the diseases are, we will be able to really get a handle on this whole system going forward. That's what my belief is.
Yeah, so we're adding one piece to the puzzle that we didn't have, and that's actually the measurement of actually, in this case, the pathogens and what pathogens you have, and call it on the benchmark scale, potentially what threshold or concentration they are on the average to the rest of the marketplace you're working in, right?
And when we look at this environment, something like crop intelligence, which is developing across Western Canada, gives you that environmental condition situation. So those are the basic principles behind this. Now I just want to take you very quickly through going through the actual data that you will get back when you enter the portal. Now there's a launch portal, there's training that or folks in Ames and across the US that help you get into the site. You get registered so that you will get the bags sent to you so that you can send in the samples. The whole shipping process is up to speed. And maybe Craig, maybe you want to review that, because that I think for guys that are watching this, guys and gals that are watching this, I think it's important to know how that system works in getting the sample to them.
Yeah, we've call it learn the process ourselves and our benchmark customers that have helped establish the thresholds across the country this past fall or through this early winter. Trace actually, and these next few slides is really digging into the TraceView portal when you actually get analysis back and how to make use of that information. But I always say this, we spend all our time talking about the agronomics and we love the science behind it all, but there is the logistics of getting it done. And, obviously we're in Canada and we're shipping samples to the United States. The lab is in Ames, Iowa. We're leveraging courier companies here today that allow us to send samples south. All that can be done ahead of time through the TraceView portal. They have a pretty good system for actually getting grower information entered, grower fields entered, and then, creating submissions to send that south. We'd send you the bags. You print your forms off the TraceView portal to accompany those bags. But I do think it is a system that is quite user friendly once you get used to using it. There's opportunities to upload your grower information into the TraceView portal ahead of time using CV files to get that information in there. So we can preload that information ahead of time as well, which if you're going to import a lot of growers and a lot of fields, that's probably something we'd want to do before fall to get ready to sample those fields. And I think it's, we have a bit of runway right now, but it'll get chewed up pretty quick as we get to spring and then busy with summer. But our goal is to be ready for a full-on commercial launch for, call it July 1, where our folks that want to work with us to get doing the trace complete samples, we want to have those farms, those fields, ready to go. And I think once we get that set up correctly up front, then the rest of it becomes a lot easier to navigate. And like Mike said, once that sample information's in there, it stays in there for the rest of the time. And as they come up with a new pathogen or they find a new beneficial organism or a phosphorus solubilizing bacteria, it'll just update that existing test that's sitting there in storage, which is pretty cool.
Okay, we'll quickly go through sort of, I captured it. Just go back, Craig, for a sec.
Okay.
Yeah, when you enter the portal, it always looks the same. And I just point out some things here in the front. These are all active buttons here. And so you can see that these are orders. So it lists all the orders that came in. And then we get all the reports here. So here's the chemistry button. If you push on that button, you get that chemistry report. So all of these buttons work. So on with the next slide. And over here on this side, you got maps, you got analytics, and we have over here analytics, and we'll get to analytics. So all you're looking at are these series of buttons and these series of buttons here, and you can move around in all of your data, okay? So here's the seed one. So you're allowed to have, or when you set up, you can select how many crops you want. In general, the maximum you can have is 6. That will be commonly showing up, because that's why we grow in Canada largely. There are a few crops, so you're not interested in looking at olives or sugar cane or anything like that. So you can mount those there. So because we have all the DNA, all you do is you look at wheat, you click wheat here in your crop button, it tells you the 6 major diseases that the DNA tells you are there. And then it lists a whole bunch more that are at these levels. It only gives you basically the 6 major ones, and then it outlines all of these that are also in this middle category that you should look out for. If it gave you all of them, you'd be just swamped. So it picks out the highlights. And you'll notice here, it's a national benchmark. So we have enough samples that we have a national benchmark, and all of these are showing them they're in the 90 percentile. And the number tells you it's 134. Oh, you can't see my cursor. If you look at the top one here, or the second one, because my eyes aren't as good, the second, there you go, 149. That's 149% of the national benchmark in Western Canada from all the samples we have for Septoria. With common bunt at the top one, it's massive. With ergot, it's 115% of the national benchmark. That's what those numbers imply. So you want to change crops, you just click under crops and you change crops to peas. Then you get all the diseases of peas. And you can see that the last one here, Ascochyta, was at 0 basically. So there are no more diseases showing because they're, nothing serious. These are ones there, but they're not showing up that are also common. So you can take a look at Aphanomyces root rot, which is of course a big concern for you, for everybody. And we have national benchmark here, but it's not giving us the numbers on the percentage yet. So that tells you that it's not fully filled out yet. In other words, the national benchmark is not totally determined, but it tells you the levels in terms of that risk as far as we can tell. Next one, change crops.
Yeah,
again.
so if you're a consultant or an agronomist or a grower and you're looking at this and you're saying, this is a particular field, I'm trying to figure out what I'm growing. If I'm growing wheat, and this is the seed selection guide, it's kind of saying, here's the pathogens that are, at whatever levels they're at compared to the benchmark. If in fact you're building out a fungicidal seed treatment, you would probably want to know this information to make sure that these pathogens are covered with that fungicide. If they're not, chances are you're either going to propagate that disease, or that disease, if the disease triangle lines up, could actually then cause harm for that wheat seedling if your product of choice isn't covering one of these pathogens. It's in the top 5 or top 6. Not to say below you see other diseases that are not high on the list, but they're still detected. They're still detected in that sample. There's a DNA sequence that hasn't been detecting take all or Pythium root rot or anthracnose or Fusarium head blight. They're still detected, but they're not the top ones in that sample.
Just leave that there for a second.
No.
Take a look here. You see this gray leaf and a leaf spot, which nobody's sort of heard of, and it shows up more in the eastern United States and eastern Canada. So even though it says that there's, and it's in the yellow or golden, whichever color, so that means it's in that range where you want to be watching for it. But just because it shows up doesn't necessarily mean it's going to be damaging to the crop, because we think at the moment that even though we got spores and we got therefore DNA here, we're not sure it's going to be a problem, because our growing season isn't long enough for that organism to go through its cycle and actually become really damaging. And I'll mention something when we get to corn. That's another matter that at a very low level is of real concern, because of what has happened in other places. So just because it shows up, it may not be a real big problem, and that's where you at the local level still have to make some decisions and be familiar with the diseases we have here in Canada, and many of you are. But then we change to peas and we can look at diseases of peas, and then here's corn. So you can see corn has a lot of issues, and this is a sample from Saskatchewan, so it's not even from Ontario. But you can see we have a lot of sort of diseases here. We have a lot of them here, but take a look at where's tar spot, right there, tar spot. Now we have signs that there's tar spot here in Saskatchewan. Okay, not a big deal, except when you go to Ontario and you find out now that tar spot got into southern Ontario back about 5-6 years ago, and now tar spot is a real massive problem in corn. So we now have information that tells us, before we didn't know that there was even any tar spot around, as far as I can tell. We have information now that says we better watch for tar spot, even though it shows low, at very low risk potentially, because it's a low inoculum level. But if we have the conditions over time for it to grow and expand, we may have a serious problem. That's the kind of stuff we can use this for, where we have identification of these and they're not even here, but we can now be alerted to watch for them.
Yeah, in this case, and this is a common practice in the U.S. now with growers that are using trace, say in this particular field, though it's from Saskatchewan, we're identifying Goss's wilt as a pathogen that's kind of it's there. It's not above the benchmark, but it's there. If you did nothing and say, well, you know what, there's actually some Goss's wilt varieties like that have some resistance. Should we grow it or not? And you say, well, no, let's not grow it. 3 years from now, you come back and test this field, and Goss's wilt is now in the upper percentile, the 90th percentile, and maybe because you did nothing about it, whereas you could have selected a variety that had some resistance. And that's what's happening is, you make the decision to protect yourself or not to propagate that pathogen on a plant that's a host to build Goss's wilt, right? So it's those types of decisions. Now that you know what's here, what are you going to do about it?
Okay. Next one. Okay, comparing data sets. You got a lot of fields. Well, if you're trying to make a decision, here on blackleg, for example, in this particular case, you can tell which fields have a lot of inoculum. And this field is probably where you don't want to, put that crop. Just that simple. Because see, I didn't point out, we'll look at, there's another button here that always shows up, and that's nutrient cycling pathogens. We're in pathogens and then chemistry, so you can always go back there. Give us another one, Craig, unless you've got a comment here about blackleg.
Yeah, same thing. If you're comparing all your fields side by side and you're looking at your rotation, you say, well, maybe it doesn't make sense to put canola on the fields that are in the upper percentile, or instead of me just programming canola that gives me the best genetics I can buy, maybe on that particular field, I'm going to find the best variety that has the best tolerance to blackleg. You'd say, well, now you've made a decision that could be the difference of 10 bushel an acre on that field. You say, what is that worth when things are tight? And now you have more information to pick the right varieties, not change the rotation. You're going to keep it as canola, but you're going to change and pick a variety that has the highest tolerance to blackleg, knowing that field is probably, it's 3-4 times higher than the rest of your fields.
Yep.
Same thing. This is club root.
Same concept of club root. You stay away from the field. Or the other thing you do is you start looking at these fields, because you'll know which ones they are. So now you're alerted. You know you have club root. There's a lot of people we're finding don't think they have club root. Well, I'll tell you what, they're being surprised. They're being surprised. So now you know which fields to look for. So where do you look? Well, you know that club root usually enters the field, and you'll find it easiest at the entry to the field, where you're putting in your seeder, your cultivator, whatever you're doing in that field. So you now know to start looking in all of those fields for signs of club root.
Yeah. So you're creating a metrics in amongst your own farm, or in this case, it could be a consultant that's looking across fields across his region or her region and saying, yeah, I'm finding this so happens these fields are whatever northwest of me, and that's an area where it seems like it's propagating. We better do something about it in that area. We better make sure if we're growing canola, we're picking varieties that have some background level of tolerance. Otherwise, 3 years from now, these numbers are going to be far higher because we start to propagate that pathogen.
The other thing that the farmers and agronomists are going to learn on all of these things is, okay, I've got, I know that I have a range of levels of infection or inoculum in that field, because this is in parts per billion, so it's quantitative. So now I can start saying, okay, over time, I'm saying, well, we had weather conditions like this. Oh, I saw a real expansion, or I didn't see an expansion. Well, over there where I took a sample, then, with a lower spot, man, we had a lot of problems over there, but now I know where to look and I should be examining it. So I think there will be a lot of learning using this as a base. If you're alert and you're recognizing where you might look for it, you'll learn how the environmental conditions year in and year out affect the incidence that it shows up as an actual damaging level, is part of my, my belief. You'll be able to figure it out. This is Aphanomyces, I think. My site isn't as good as it used to be.
Yeah, same thing. Here, I would go the opposite. You'd say, well, there's a number of fields that are actually not detecting Aphanomyces, obviously, or say we did 3 zones per field, we're not finding it. But in all reality, now you make a good rotation decision. Or over here, you could say, I can grow peas on peas, and you'd say, well, that's taboo. But if your greatest concern is disease, and we're saying you don't have it, then what is your risk, right? Whereas over here, on the left side, you say, well, these fields have a background level. It may or may not impact the plant, but we know it's there. So we should probably be doing something about it at minimum. Maybe that's extending the rotation on those fields because of the levels that are present.
Right, and with both Aphanomyces and clubroot, they're both swimmers. They're flagellated, so they move around in the soil. So that it takes moisture, and you'll soon learn that in a moisture you're going to have a problem, or where you have a low spot you're going to have a problem, and maybe you're going to take those out. I mean, there are a lot of things you can consider doing. Plus, you can go back and see if you still have inoculum in the soil at high levels. Trace phos. Okay, we get a phosphorus, and this is a really good one because these are 3 samples from a field based on elevations. And if we go here, you'll see that these soil tests, we give you all 3 ways of looking at phosphorus. And Craig, you got more experience in terms of analysis. So we get the Bray, the Olson, and Malik so you don't have to worry about which one you have. You won't get the Bray P if the pH is above 7.4, I believe, because you just won't get an analysis. So you won't get that Bray at a high pH. Then it gives you an outread in terms of saturation. Another, what percentage of cation exchange is loaded? In this case, it's 56%. It's pretty medium, so probably the most you would want to do is apply enough phosphorus for uptake. What's the term, Craig? For
maintenance.
Maintenance. That's the term. So you might just put a pop-up and a little bit down there just for quick release, because phosphorus doesn't move a lot in cold situation. This is going to be changed. It's called liberating agents. But if you have a low saturation of phosphorus, for example, we're down here, and you put down a lot of phosphorus, you would probably want to use a chelating agents. That's going to likely be changed to chelating agents, because if you have next to no phosphorus in the soil, anything you apply will be tied up pretty quickly. So you may want to put a chelating agent in there, and you can make that decision. This has to do with biology in terms of solubilizing microbes. This is low. This is a low level. So if you have a fair amount of phosphorus, which is not bad, you may want to put a microbial application or some sort of biostimulant that would help to sequester some of that phosphorus and release it in the cycle. And this is stability? No. Yes.
Suitability for build. No.
Yes, suitability. Well, you have some room in there, so you might do a little bit, but not likely going to be a big deal considering you're fairly saturated. This is
that's where I find it interesting, because phosphorus is one of our greatest challenges in Western Canada and Canada as a whole, North America as a whole. If we want to move the bar on crop production, we've got to find ways to get more phosphorus into plants. But in this case, if all we had was parts per million, for the most part on the field average, we take the three zones, say at the Olson at 13, it's not parts per million. It's not terrible. Like, it's not deficient. Saturation is actually pretty good. But maybe one of the challenges we didn't know is that we're lacking some biology, the organisms that actually convert it from an unavailable pool to an available pool. So by actually adding biology, we could actually improve the phosphorus uptake into the plants by creating more available source of phosphorus, right? So it's that kind of information, and just conversations we've had since we've been benchmarking, people testing fields that have been manured and not figuring out why phosphorus is still moderate to low in tissue tests. And it could be partly because we're actually missing some biology in the soil that help convert that to an available source, right? And so that's, to me, when we look at phosphorus at a deeper view, the trace phos gives us that extra insight to understand how we actually get more phosphorus into plants.
And this is the main one is an average, but if you look below, you can see that in this case there were several samples, 3 different zone samples in there, and you can look at the variability across your field. You can see that in, say, zone 5 and 6 here, you can see that the biology is just about nothing in there at 10. So that would be a site where you say, okay, on a variable rate basis, if you're going to, we're taking this to sort of theoretical points of view, you can see in also zone 3 and 4 that the biology is really, really low. Give you some options.
Yeah.
Analytics. Go ahead, Craig.
No, go ahead, Mike.
This is the bar along the left side here, as you can see, and these are all the analytics you can utilize, and it'll draw you up some analysis on it. And I'll just show you, I think I go to this mycorrhizal, for example. It'll give you, there's your mycorrhizae. It'll give you levels of rhizobium. So it's not just looking at the disease components, it's looking at the beneficial components as well, like mycorrhizae in this case. Next one. You showed this earlier today. This is nitrification potential. So you can see that these fields have a lot of potential for nitrification. In other words, the conversion of ammonium in the springtime to nitrite. And nitrite is sort of the key step in terms of the process of nitrification and regulating that, and then at the end you get denitrification. So you're saying, okay, if I've got a high risk of nitrification here, guess what? I probably should use a nitrification inhibitor on my fertilizer that I'm putting down, my urea. That'll handle that. Now at the present time, and I'll just spend a minute here, at the present time, we're not measuring urease. In other words, the whole nitrogen process of utilization of nitrogen from urea goes to ammonium, initially to ammonia, and we can slow that up by applying stabilizers to reduce the effect of urease, the enzyme. Urease, which is used to break down the urea, is produced by microbes in the soil. Very shortly, we will be giving you a level of urease potential in that field. And the whole nitrogen component, including nitrification, will move out of analytics, and it'll probably be like a FOSS. It'll be specific for nitrogen going forward, because government is really concerned about denitrification. So on these, you would put on a stabilizer. You would probably put a stabilizer on all of these, and you'd say, well, I haven't got much potential here, and I need to save some money. I'm not going to put on a stabilizer there potentially. Greg, you maybe want to make a comment here?
Yeah, we're at our time pretty well, Mike. We'll spend another 5 minutes, but yes, when we talk about government programming versus agronomics, this is agronomics at the field level, where I always say technology will outpace policy, and in our world, we're now saying we're going to make growers better managers of nitrogen. So nitrous oxide losses should be lower if we manage nitrogen on fields that are more susceptible to loss. But having this information, not only are we going to improve the agronomics, hopefully get more nitrogen into the plants, we are actually appeasing policy at the field, per field by field, not generally speaking across a region or a farm. It's done right at the field level.
Okay, next one. This is just showing the fungal to bacterial ratio, because we want more of a fungal one, and we can skip to these quickly here just to show them what data comes out of this automatically. Field discovery, we want to do some examination of the field. For example, we want to look at clubroot in canola, and we want to look at the effect of pH. Next one. So we can set up criteria which will search it. So then we can see here, let me get rid of this one. Oops, hold it. What happened?
Yeah, part of this is called creating rules. So inside a TraceView portal, you can actually start building your own rules where, we want to compare clubroot to soil pH. That's something that people believe is to be true. Now we can either prove that out or just dispel it basis the science by combining pHs and clubroot infection rates across fields. And that's the beauty of this technology is, what is the metrics that says Aphanomyces and Fusarium root rot are tied together? Well, now we can create a rule where we put those pathogens side by side, and we can look at the fields that are greatly impacted by both or not. Maybe there is no correlation there, but now we're just basing it on parts per billion for each organism per field.
Exactly. Next one. Ergot. They're just looking at ergot.
Yeah, go ahead.
Just move on to the next one. We're going to take a look at ergot here. Okay, ergot in spring wheat. Ergot and copper, the relationship between ergot and copper levels, because it's believed that copper really influences this. I'm a believer it's important, but so do the environmental conditions. So you can take a look at this. Copper levels here, ergot levels here. You can see at 0.15 parts per billion, we have a very low copper level, which sort of shows the relationship. But at this particular site, we've got virtually the same thing, same levels, and it's got a high copper incidence. Well, I'm not sure what that means, but in that circumstance where we had a high level of inoculum, we might have had really good conditions for the infection to take place. I don't know. Local level, you'll know. Next. Maybe skip to
so you have the Yeah,
Go ahead.
you have the ability to map out your farm, map out a region, look at nutrient cycling. So this is phosphorus saturation levels. So if you actually create borders for your fields that you sample, essentially you can visualize the impact of a pathogen across the landscape or across your farm. You can look at nutrient cycling across the landscape or across your farm. If you do it by zone, you can even add that much more detail to a field. So the visualization of everything that we just looked at is all possible through the TraceView portal. This is obviously a Google Maps, but by having field boundaries and GPS points, it allows us to actually paint a picture of what could be a concern or what could be a trend across your landscape when you look at certain aspects of the analysis.
These are the last two slides. I just threw this in here. This is rhizobium for peas and lentils and so on. But this is a large number of fields. Take a look at the variability. Each one of those lines is a field. That's the kind of analysis, say, an agronomist can do or we can do across Western Canada. So how do you make a decision which ones you would inoculate again? Right now, you just inoculate them all. Well, maybe you didn't need to inoculate them all. We don't know. We will learn in time. Next one.
This is a little bit, you think green is good, but green is low in this case, right, Mike?
Rhizobium levels, no, not this one. I don't believe that's the case. The green are high levels.
Okay.
The rhizobium in this particular.
It'd be the lower percentile. The red would be the higher percentile.
Sorry, that's right. Yeah. That's right.
Yeah.
And here's your total mycorrhizae, and you can see how low the benchmark is. So there's tremendous variability. We'll learn more about this in time because we have treatments that we can use to increase these levels.
Yeah, I think the point you want to make here is fields are not the same. When you look at beneficials like rhizobium or mycorrhizae, these are organisms we absolutely want, but it's hard to find fields that are the same. When we test across a landscape, there is a high level of variability. So knowing this information is going to allow us to make better decisions on a field by field basis.
Exactly.
Yeah.
And this is just touching it. So here we go into the end of it.
Yeah. So yes, lots of information. Obviously, we're over time by a bit, but if we end where we start, and you ask the question, what if you had access to this information? What if your ability to make decisions is now at an all-time high, from rotations to seed selection, picking the right variety, to managing in-season disease, to better understanding nutrient cycling from phosphorus to nitrogen, to even new nutrients like potassium. We're going to look at potassium solubilizing organisms moving forward. Would you go back to not knowing? And I would question everybody we have talked to date that's worked with us, growers I've talked to in the US that are using it. I have yet to find somebody that says we're going back to not knowing. We want to go back to not knowing what's going on in our soil from a living aspect. And so I truly believe agriculture will never be the same once you engage and once you start leveraging, the DNA aspect, the living aspect of your soil. You are not going to go back to say, I don't want to know. You will want to know. And part of the reason why you want to know is because Trace actually makes it easy to know. The TraceView portal is a tremendous tool. The AI of the portal allows you to simply find what you need to leverage in your operation to make better decisions. And for us, that's as integral as the DNA sequencing itself, because you can actually make use of the information. So any closing comments, Mike? I mean, that's a lot of stuff in an hour and 10 minutes. And we're just kind of introducing it to the market, but we want you to be.
And I can tell you, up until 6-8 months ago, I'd never heard of metagenomics, and it wasn't until about 3 to 4 weeks ago that I actually had access to looking at this data. So my learning curve has been pretty rapid, and I'm not a computer whiz. So for farmers and sort of agronomists who are maybe a little bit older who are scared of this stuff, it's like anything. Once you get into it, you start learning, and the beauty of this with Trace is prepared to support you all along the way to help you get up to speed. That's the beauty of the whole system.
Right on. Well, thanks for coming on, Mike, and I know you've spent a tremendous amount of time, as this is your nature, to actually, you never part way in, you're all the way in. So you're all the way in on metagenomics and understanding, the living soil, and now you have tools that are going to keep you engaged for the next 10 years of your life, which is amazing. Fantastic. So if you want the CCA credits, again, go to the CCA, download the CCA app, and you'll be able to find the Trace Genomics presentation from Taurus. And if you want to stay informed, you can sign up for the Taurus newsletter by taking a picture of that QR code, and we'd be happy to keep you up to speed. We will be pushing this out socially for those that registered. I know not everybody can get on a Tuesday morning after a Monday holiday, but we'll send you this presentation directly as well. So thanks for coming on, and enjoy the rest of the winter or third spring, whatever you want to call it, that's coming our way. Take care.